Welcome to Lecture 6. We will be picking up with neurotransmission and finishing up what an action potential is and talk through some of the finer points that you hopefully will gain understanding about. So the action potential, another word for that is a nerve impulse, and basically what that is it's just a series of action potentials that are occurring, or the propagation, that's the fun jargon term. Looking at this picture, this kind of gives you an idea of what's happening, right? There's this certain polarity, right? It's negative inside the cell and it's positive outside the cell. And when you have an action potential, those reverse temporarily, and then they go back to being the way they're supposed to, right? So they return back. And this is basically what this is showing. It's just a nice propagation. Each portion of the cell membrane is having this type of response because the positive charges are flowing in next to it, which causes the sodium channels that are near that to detect those, and then they start to open and basically just causes this flood and this propagation like a domino down the axon. The refractory period, that's that short period where the neuron cannot send another action potential, and that's primarily because of that hyperpolarization that occurs where the cell becomes more negative, and it takes a lot more of an effort to actually create. It's very hard to overcome that large negative deficit to get to the -50 to have the action potential, right? So there's the whole design behind it is to really protect the neuron from excessive firing. The other thing that goes on is that you have to understand is that the size and shape of an action potential is constant. That means that the amplitude from here all the way up to +30, and then this hyperpolarization, that does not change. It does not increase. It doesn't become less. What happens is as soon as you hit the -50, you're going to have a full-on, it's an all or none law, which means if you hit the -50, it happens. There's no going back. There's no turning back. I talked about a toilet last time. Another thing you can talk about are dominoes. Once you hit that first one, they're gone, right? Okay. So the speed of the impulse, it does vary. And so we're not talking about the intensity of the action potential, but the speed at which it moves down the axon can vary. An unmyelinated axon is one that has none of that fatty insulation wrapping that is created by the oligodendrocytes in the CNS or by the Schwann cells in the periphery. Just to touch back to past classes and some terminology. So this is what that photo looks like. That's what this is describing. It is showing us what a action potential on an unmyelinated axon, or what a nerve impulse, would look like on an unmyelinated axon. Every portion of the axon is having the change in polarity. Now, myelinated cells actually move more quickly. And the reason why is because it doesn't need to have an action potential sequentially right next to each other in a series, right? And that is because the myelination, or the fatty substance, creates a insulation which allows the sodium ions to move down the axon in the direction we need it to the next node, where then it causes basically a booster or a burst of more sodium coming in, which then causes more sodium to rush in, and it moves down its concentration gradient. It moves away from itself, and then it creates another boost, and it just keeps jumping. It's basically a jump. Saltatory is when you break down the word, it means to jump. Here's a picture of that. And instead of, as you see here, you have the exchange happen here, the sodium starts to pass down this way, causes these to become, get into an environment where it's hitting positive or -50. So there's a rush in of sodium, but it jumps over to the next one, and it literally is causing an explosion or a little booster each time it jumps over, making it a much faster propagation down the axon. Here are two fantastic videos. Both are linked on the page that says helpful videos. It is below my videos. I have 3 videos there that kind of hit on specific topics that you guys really need to know. So go check out those videos now. Pause me, and I'm going to come back to me, and then we'll pick up.
Hello everyone, Dr. West here. So I'm actually not going to show you both videos that Dr. Bennett was just referring to, because I actually already showed you one. I included one of the two videos that she was just talking about in the previous lecture. I guess I'm a big picture kind of person. I like to see stuff ahead of time. So in the previous lecture, I showed you the video of the action potential where it was showing the channels that were opening, the sodium and potassium channels, as well as the pump. And they sort of had the cutesy animated version of the two channels that were opening and closing. And then they said, let's do a recording now. And then they had the cheesy animated recording thing. And again, it was doing that. So that was the first one. If you want to go and review it, I highly suggest that you go back and review that one. But I am going to show you this one about what is meant by saltatory conduction. But essentially what it means is just that the myelin that we talked about in a previous lecture. So if you remember, we talked about glial cells. And we said that there are two types of glial cells that will wrap the axon with myelin, and that this is important for a lot of reasons. First of all, it helps protect the axon. It keeps it from getting damaged. So just like we don't just have bare wire lying around whenever we are trying to conduct electricity to our pieces of equipment, like our TV or our computer or whatever it is, we wrap our wires usually with some sort of rubber or plastic. It's a similar kind of concept for the axon, right? It's wrapped in this fatty substance that's made by the glial cells. In the case of our body, it's either a Schwann cell or an oligodendrocyte, depending on whether it's central nervous system or peripheral nervous system. But nonetheless, this actually also serves a secondary purpose because it speeds up the action potential, because instead of having to produce that opening and closing of sodium potassium channels and having a sodium potassium pump that uses up all that energy every single spot along the length of an axon, all you do is you have to have that combination of stuff at just the little tiny gaps in the myelin sheath, and we call those nodes of Ranvier. So again, this is what we mean by saltatory conduction, is that you basically have little mini recreations of that action potential at each node of Ranvier. And that's what's meant by saltatory conduction, that it almost seems as if the action potential sort of jumps down from spot to spot to spot, so from node to node to node, down the length of the axon. So that's what you'll be watching in this quick video. So hopefully that makes sense.
What you will see and hear in this presentation is the development of myelin in the peripheral nervous system and the propagation of the action potential along a myelinated axon. This multimedia presentation will be most helpful if you already have a good understanding of the Schwann cell and the electrochemical process of the neuron, called the action potential. The Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage.
She mentioned it very quickly here, but she said it was made up of lipids. Lipids is a fancy word for fat, right? So this is what I mean by it's a fatty substance. If you remember way back to a previous lecture, right, and if you've ever looked at any kind of meat, even if you don't eat meat, but if you go like to a grocery store and you look at the meat case, right, the fat on meat looks kind of whitish. That's why we call it white matter. That's why the axons look white, because they're surrounded by this fatty substance made by the glial cells. Anyway, I'll stop interrupting. Go ahead.
Advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the myelinated axon. Here is the neuron, and you can see the repeated Schwann cell membrane forming the myelin. Note that there is a small space between the Schwann cells where the axon is not covered by the neuroglial cell. These spaces are called nodes of Rampier. From what you already know, action potentials occur at the axon hillock and continue to be repeated away from the cell body, much like dominoes falling one after another. An action potential starts on a polarized membrane, which is -70. A stimulus causes the sodium gates to open slightly, and sodium starts to trickle into the cell. If the cell reaches -60 or threshold, the sodium gates open wide and sodium floods in, bringing the inside of the axon to +30. At this point, the sodium gates close and potassium gates open. Potassium starts to pour out of the cell. This allows the neuron to become polarized again. Then the sodium-potassium pump starts to actively transport sodium out and potassium back into the neuron. First, we will look at the propagation of the action potential in the unmyelinated axon. Propagation is the repeating of action potentials down the axon. The action potential is repeated because as the sodium comes in, it diffuses to adjacent areas within the axon. As the sodium increases in this area, threshold is reached. Sodium gates open wide, sodium rushes in, causing depolarization and an action potential. As the sodium enters this area, it diffuses through the axoplasm and another action potential is created. This continues down the length of the axon. Now look at the myelinated axon. The same process applies to the myelinated axon. An action potential develops, and as the sodium comes in, it diffuses through the cytoplasm of the axon. It continues to diffuse through the portion of the axon wrapped in myelin. The increased sodium concentration reaches the node of Rampier, increases the axoplasm to -60, and depolarization occurs. The sodium gates open wide, sodium floods in, and we have an action potential. Again, the sodiums diffuse through the axoplasm, reaching the next node. An action potential develops. The process is continued down the myelinated axon, passing from node to node. Compare the unmyelinated axon with the myelinated axon. You can see that action potential reached the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.
All right, great. Now that you've seen the slow propagation and you've seen the fast propagation, and it really talked a lot about everything in that action potential going on there, and I think those are great videos to help really drive. And if you noticed, one of those videos mentioned that threshold and hyperpolarization and resting memory were different millivolts. Make sure you pay attention to the ones that I talk about in my lectures as well as in the textbook. Those are the ones you have to know. So when information is coming into a neuron, one of the things we've talked about is we start to lecture and I start to share with you that it's one neuron, one dendrite receiving information from one axon, and that's because we're trying to understand it in a simplistic manner so that you guys can wrap your heads around it, as it's primarily, for most of you, new information. What you need to understand, though, is that neurons have thousands of connections, and they're receiving input from thousands of axons, maybe tens of thousands of axons of other neurons to create messages and communication. How does then that neuron actually interpret all of these stop and go messages? Because remember, there's only two messages that a neuron can receive. It can either receive an excitatory message or an inhibitory message. Excitatory meaning that the sodium channels are opening and sodium's rushing in and the cell's becoming more positive. It can also receive chloride, can receive an inhibitory message, which causes chloride ion channels to open up and allows chloride ions to come into the cell, which then decreases. And it's this combination of multiple types of messages that are coming in. It turns out that there really is only one place that actually matters in a neuron of what its polarity charge is for an action potential to occur. There's just one place, and we're going to talk about that in a second. But basically what happens is all the information's coming in that dendritic tree, all the different dendrites that are out there, information's coming in, and that is going to, based on concentration gradient, those sodium and chloride ions, maybe the potassium ions, or whatever other ions might be playing a role, they are following their concentration gradient. And if you think about it, if all the information's coming in from the dendrites, the cell body is lower density, is a lower concentration of all those ions. So the ions start to translocate. They try to start moving towards the cell body. And this is where all those kind of informations converge. So how does it actually incorporate all of these pieces, right? So let me talk to you about the two different messages so that we're fully on board with it, and then we're going to talk about where it's important. All right, so there are two messages, right? There is excitatory, postsynaptic potential. That is what is happening on the neuron that's receiving the information. So postsynaptic means after synapse. So this is what's happening at the dendritic level. The message can be excitatory or inhibitory. So an excitatory is a brief depolarization, which means that sodium is allowed to influx, or come inside. Then there's the inhibitory postsynaptic potential, which is a time point where the cell membrane becomes more permeable to potassium or to chloride. What happens with potassium is that if potassium channels open, they will leave, they will rush out. Think about the concentration gradient, right? It is sending the message that you want to be out of the cell. You want to be out of the neuron because there is more potassium inside the cell. The other thing that can happen is that the chloride channels can open. And if you go back to the previous lecture talking about resting membrane potential, there is more chloride ions outside the cell. Therefore, when those channels open, they're going to come rushing in. And that creates a more negative environment inside the inside the neuron or the postsynaptic neuron, which creates a more negative environment, meaning that there is less likely to be an action potential. So here's a quick picture just so you can see what we're talking about and looking at it in the millivolts. So an excitatory or a positive message causes sodium channels to open, which allows the millivolts to go up. Inhibitory allows the chloride channels to open, allowing chloride to rush in, reducing the polarity, or allow potassium to rush out. Again, net negative charge, more negative charge occurs. So those are the two messages that you can have. Okay, so now we're going to look at how these messages are summed in the postsynaptic neuron. Those are driven by two different factors. There's temporal summation and there's spatial summation. Temporal is time related. So that's getting information at the same time or repeated messages very quickly sequentially. And this can occur by one neuron sending a message down its axon, but if you've seen some of the pictures, the axons can actually bud and create multiple different terminus, and those different terminus can affect the same neuron, sending the message at the same time, really close to each other. Whatever that message is going to be pretty potent. Let's look at a picture. So if you have wide temporal spacing, this means a message every, so let's say 5 milliseconds. You've got this nice pacing. Whether it's excitatory or inhibitory, it's not going to do much for you. It's not having much of an impact. Now, if they're close, getting closer, let's say this was 1 millisecond and then 2 millisecond, 3, there's the next bump. Look how much closer that neuron's getting to threshold. Much more likely to happen. Again, if you're getting the negative impulses relatively close, you're going to have a substantially less likely chance of having a threshold. However, if you get that message at the same time, your chances of reaching threshold are far superior than if they were separated out by 5 milliseconds. So that's what we're talking about, is this timing. And this is true about the negative too. If you get two negatives in a row, it is, or right at the same time, simultaneously, two different neurons or two different synapses, then that is a much more potent message than if they're separated.
Hello, Dr. West here again. Wanted to break in one more time and just let you know a simpler way, at least in my mind, that I remember temporal and spatial summation. So as Dr. Bennett has just been talking about, temporal summation is when you get several impulses or several bits of information at about the same time. So basically those dendrites, or even the cell body, is catching neurotransmitter message at about the same time, right? And so that makes it seem like, whoa, this is a big deal, right? So the image that she was showing you, right, could work for both excitatory as well as inhibitory, right? So if you receive one message, right, the pump is going to have enough time to have returned all the ions to the correct location. But if another stimulus comes in, right, again, the pump's going to have time to fix it. But if you get a stimulus and before the pump has had a chance to fix everything, you get another stimulus, you get even closer to threshold. And so you're a lot more likely, right, to be able to produce an action potential because you got closer to threshold. And sort of the opposite is true for an inhibitory signal, right? One stimulus makes things go a little negative, and it's not exactly the same pump because we're not exactly talking sodium potassium, but the similar idea is that eventually your cell will stabilize itself back to where it wants to be. If another stimulus occurs, again, it will stabilize itself. But if it hasn't yet stabilized itself and then you get another inhibitory signal, it's going to become even more negative, which will make it even less likely that it'll fire an action potential. So I'll give you an example. I'll start here and then I'll show you my silly example of how I remember what this one is like. But for an inhibitory postsynaptic potential, as far as temporal summation goes, this would be like sort of like looking at your bank account. Like, do I have enough money for this? Or how's my bank account doing? Well, you get a bill in, all right, so your money goes down a little bit, but you keep working. All right, I'm good. I'm still good. I'm still good. And even if you get another bill later, you're probably still good, right? But if you get two bills back to back, whoa, now you're really going to be in a hole. And you're going to have to be thinking about, like, am I going to be eating peanut butter for a couple weeks before you have a chance to kind of fix it? So that's kind of what temporal summation means, is that if you get things too close together, timing-wise, right, if everything comes in all at the same time, it's a much bigger deal. So here's my little silly example for EPSP. See if you remember this character.
Lewis, Mom, Mommy, Mama, Mama. What? Hi.
Yeah? Does that make sense? Right? Mom, right? If it was just one mom, it wouldn't be that big of a deal, right? Mom, no big deal, right? But when it's mom, yeah, it's much more annoying, and it makes the mom go, what? Yeah, all right, so that's how I remember that one. So hopefully that will help. And I said, I'll bust in just a few more minutes and I'll talk again about spatial summation.
The other one that we're talking about is the spatial, and this is talking about location. When we look at it, I'm going to show you a picture in a moment. So when you think about dendrites and you think about how they're outlined and laid out from the cell body, there are tons of them, right? Lots of places for that neuron to interact with other neurons and be stimulated. It's the whole point of dendrites, is to collect information. The chances of a neuron receiving input from only one neuron at a time and only receiving one message at a time is highly unlikely. So I'm going to show you what can happen spatially, whether information's coming in all from one direction, or if it's coming in from two different directions, or if it's close to the cell body, or if it's way far away from the cell body. So location matters dramatically about how much this actual message is going to affect the neuron's communication beyond itself. Here's an example. A is having them on two different portions of the of the neuron's dendrites sending information, not as likely to reach threshold. It could, but it may not. But if you get them both from the same side at the same location, you are going to have a much more potent effect on the relative polarity of that neuron in that area. Hope that makes sense. It doesn't get diluted. It's more intense.
Hello again, Dr. West here. As promised, I'm going to bust in and give you how I remember the idea of spatial summation. So the idea here is that you're not just getting input from one source, you're getting input from a lot of different sources. And it's because of this sort of additive nature that it's a lot more likely the neuron is going to produce an action potential. And so how I remember this is I sort of think about like all the different horror movies or whatever that you might have seen, right? There's always one scene in the horror movie, and think of any of them like Godzilla or like any kind of the movies where aliens are attacking or cities getting blown up or whatever it is. There's always one scene where like everybody's running away and the whole town's like freaking out and they're like, they're all running away, right? And there's always one person that comes out of a building like, hey, what's going on? And they look around and they see, whoa, everybody's running. Maybe I should run too. And they start running, right? That's kind of how I think of spatial summation. So the idea would be if you were to go out into the world, and again, you just see one person running, you might go, hey, that's kind of weird. But you probably wouldn't think, you know what, there's danger. I should probably run too, right? You'd just be like, oh, that person's running. You may not even notice them, right? It may not even cross your threshold enough to even notice it because, again, it's just one person, right? But if you went out and all of a sudden, like, everyone was running, especially if they were, like, also screaming as they were running, you'd be a lot more likely to go, whoa, this is probably important. I should probably do the same thing. I should probably start running and screaming, right? That's kind of the idea with spatial summation, is because if a whole bunch of people are all doing it, right? This one is doing it, and 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 this one is doing it. Everybody is all doing it, right? You're a lot more likely to go, yeah, it's probably important because a whole bunch of people are doing it, right? In this case, the neuron is going, all right, this is a probably important message because I'm getting the same message from this axon ending and this axon ending and this axon ending and this axon ending. I should probably send it on, right? Because the cumulative effect reaches threshold. That's kind of what this is. Anyway, that's how I keep it in my head. Hopefully that will help you remember it too. Happy studying.
Now, you might be wondering, well, who cares? You know, I mean, in all honesty, we could have out here it be positive like 100. But a neuron doesn't care what its far-reaching dendrites are doing unless it's potent enough to make its way quickly down the dendrite because the concentration gradient is so high that it goes on to affect the more important area. So when you think about the action potential, we've talked about the role of voltage-sensitive channels. At this point of all my talking, we have not been talking about voltage-sensitive channels. We are talking about ones that are sensitive to neurotransmitters. For an action potential to occur, though, you need to be in a place where the voltage-sensitive channels are present. And it turns out there's really only one place that is important in the cell body for where these voltage-sensitive channels are, the axon hillock. So what is the axon hillock? That is where the cell body and the axon are joined. In it, we will find a lot of voltage-sensitive channels, and this is where the EPSPs and IPSPs are integrated. And if at that point you have the neuron's relative polarity reach negative 50, you're going to have the action potential be initiated. So here's a picture. Hopefully this will help bring it home. This cell body is receiving all these different EP and IP, EPSPs and IPSPs from all these different dendrites bringing information. Spatially, if the messages are coming in as excitatory in this area here, closer to the axon hillock, you are going to more likely have a depolarization here and reach that negative 50. If these four right here were all negatives, the chances that all these positives out here were having an impact here are highly unlikely unless they continually come. So if they become a temporal summation and they keep coming and it keeps sending, no, you're supposed to have a message, you're supposed to send a message, you're supposed to inhibit the next neuron, right? If that message keeps coming and these inhibitory ones start to fade, then these more potent excitatory ones are going to work. So the axon hillock is the most important part. If we get to that negative 50 here, you're going to have that action potential go down and there's no turning back most of the time. So back propagation. And I want to bring this up and just bring it to your attention because some of you are very interested in neuroscience and cognitive neuroscience and those types of ideas. Back propagation, for the nature of this course, we don't really talk about it, and I'm not expecting you to understand it because we as a field don't really understand it yet. So I don't feel like there's any point in you trying to learn something that we don't understand yet. But I want you to recognize it does exist. So I will tell you, I will teach with you, and we will work on the action potential only goes in one direction because we have that hyperpolarization, and that is true like 98% of the time. But there is a small percentage of the time where the action potential will actually go from the axon hillock to the dendritic field. Don't fully understand why we're doing that or what's happening, but it is a possibility. Now we're going to take some of this information and try to help you apply it to your life so that you have an understanding of why you're learning these types of things. So action potentials are what are responsible for allowing us to sense our world. Light hits our eyeballs into our retinas, and the retinas are photoreceptors that are picking up the light that's coming in, and then it's being sent back via action potentials to our brain to be interpreted. Our hearing's the same way, so picking up the changes in voice, sound waves in your ears to hear the words that are coming out of my mouth. Again, all action potentials. Our brains, you can shine a light on a brain, it can't do anything with it because it cannot interpret light. Our brain is only good at interpreting action potentials. So all of our senses are related to this. Let me give you some more examples. So touch. We're going to get to somatosensory in part 2, but it's a great example here. Each one of our hairs on our bodies can detect the slightest displacement. Some of us are more sensitive than others. If you think about it, some people can create anxiety and scared feeling just by watching a movie, and the back of the hair stands up on their neck, and then it causes, an unfolding of all other sensations in your body of being freaked out. Your heart rate goes up, your breathing rate goes up, all those kind of things, right? That is a very real possibility that people can freak themselves out. But what I want you to understand is that that is part of your nervous system, right? And what we have at the base of each one of our hairs is a dendrite for a sensory neuron. So when the muscles move that, or somebody else's hand just barely touches your hair on your hand or your neck, there's a dendrite present to detect that. So let's look at that. What we're talking about right here, right? So here's the dendrite that's wrapping around the base of the follicle of the hair. This mechanical movement of the hair by a feather, by your own motor neurons, doesn't matter what, whenever there's a detection of the hair moving, it causes sodium channels to open in this neuron at the dendrite, and then it causes basically an action potential. It sends that information to the cell body, which then creates an action potential up to the brain to be interpreting that you have something touching you on your hand or your neck, and that might make sense, and then there's no anxiety. But it also may not make sense because you might be sitting at your desk or watching a movie or whatever, right? So this is the body working, and this is why we have to understand the action potential piece. So again, the hair displacement causes the stretch sensitive channels to open, and then allows the sodium to flow in. Another example is how our motor neurons communicate to our muscles. So an axon for each one of our motor neurons has synapses with muscles, and so this is going outside the nervous system. We're talking about nervous system to muscular system discussion or communication, which is important. It has to do this so that we can move. The axon terminus of the motor neurons are creating a neuromuscular junction with the muscle. And it looks a lot like an axon from, any other type of axon communicating with a neuron or whatever. It doesn't matter. They all look the same. They have the axon coming down. They have the axon terminus. They have branching that occurs, and then communication, basically. So how? Because these, again, it's creating a synapse. It's creating a junction. They're not touching, right? Just like our neurons don't touch. There's that synapse. The way the nervous system communicates with the outside of the body or outside of the nervous system, the periphery, is the same way. It creates a junction or it creates a gap and it communicates. So it has to release something. And this is my teaser for the next part of class. It's communication between. So the neuron will release acetylcholine. And acetylcholine then floods the muscle, which has receptors for acetylcholine, and causes a contraction. And how does that happen? The acetylcholine binds to a receptor, causes sodium channels to open, calcium channels to open, and then those flood in, causing a change in the muscle formation, allowing it to contract. So that's my segue into neurotransmission between neurons. We're going to talk about this more in depth. So I wanted to introduce the idea. But these are the tangible ways of why this information that you're learning is important to learn.